Well, good morning. Welcome to NEUR3301. It's me, your old pal, Dr. Harry, and today we're going to learn about the basics of epigenomics. So, last class, we learned about kind of some of the finer points of genetics and genomics, and now we're going to be learning about the epigenome, which is the thing that goes on top of the genome. So, let's get started. Hope you're all comfortable. Hope you've got your coffees out and you're ready to sit down and enjoy a nice lecture about epigenomics. So, epigenomics is sort of the genome-wide instantiation or the genome-wide version of genomics. In the same way that genetics is the study of genes and their function and their modes of inheritance and their effects on phenotype, and genomics is the application of that across the entire genome. Epigenetics is the study of what you would describe as mitotically or meiotically heritable changes in gene function that cannot be explained by changes in gene expression, by changes in DNA sequence. And so, of course, epigenomics is the application of that principle across the entire genome. So, the thing that you want to kind of look at when you're talking about epigenetics is the mitotic/meiotic heritability. There's lots of things that can affect the function of genes that are not heritable. You can have different levels of transcription factor expression in a cell. That doesn't tend to be inherited across cell divisions. Epigenetics, in contrast, is inherited across cell divisions, and this means that the information carried by epigenetics is preserved across multiple generations of cells. This means that cell types that use epigenetics to maintain their cell type identity, this can be preserved across divisions. And also, perhaps relevant to our purposes, epigenetic changes that are caused by the environment can be preserved across cell divisions and potentially even across generations if they're inherited through meiosis. And so, in the last class, we learned about our old friend Conrad Waddington, and he described an epigenetic landscape that was sort of an allegory, I suppose, for the way that cell function is gradually canonized by successive decisions in cell fate. And the epigenetic landscape, as we saw, could sort of be defined by a landscape of transcription factors, active at various stages during a cell's maturation. That is sort of the old-fashioned meaning of the word epigenetics. It didn't have a terribly precise definition. Nowadays, it's been made much more rigorous. So in either case, what we're kind of asking fundamentally is, how is it possible for the same DNA sequence to produce all of the structurally and functionally different cells in the adult organism? As I've mentioned in previous classes, every cell in your body that has a genome, red blood cells don't have a genome, but every other cell that has DNA in it has the exact same DNA. And so, given the DNA codes for the genes that make your cells do the things they do, how is it possible that the same DNA can result in so many different kinds of cells in your body? And the answer is that genes have to be turned on and off in a certain way, and epigenetics plays a role in that. So, it was clear, I think, from earlier studies of chromosomes that that chromosomes were the carriers of genetic material. Remember how I talked about this was a little bit controversial at first. It wasn't clear to early scientists whether, they knew that the heritable material was in the nucleus, but it wasn't clear that it was the DNA or if it was the proteins, like the histone molecules in the nucleus. But it was eventually determined that chromosomes and the DNA that they're made of were the genetic material. But what wasn't known up until the 70s was whether a typical adult somatic cell, so like a neuron or a glia cell or a liver cell or something like that, had all of the DNA needed to actually direct embryogenesis. In other words, it's obviously an embryo would have to have all of the DNA that you could possibly need, because an embryo ultimately will differentiate into all the different cells in your body. So it's you could kind of take it for granted that an embryo is going to have all the genes. But what you may not, what may not be the case is if a mature adult somatic cell, say in your liver or your brain, still has all the same DNA. A lot of people back in those days thought that maybe what happens as cells differentiate is they actually remove pieces of the DNA that they don't need. So a neuron might go in and slice out all of the DNA that are involved in liver cells, thereby making it impossible for the neuron to ever express those genes. This was seen as a pretty legitimate possibility back in the day. So up until the 70s, this was one of the many competing hypotheses. The other would, of course, be that all the cells have the same DNA. They just turn off the genes they don't need somehow. So back in the 70s, Lasky and Gurdon showed that you actually could suck out the nucleus from an adult somatic cell with all of the DNA that it might happen to have and inject it into an enucleated xenopus egg. This is a kind of frog where the egg cells are really big and you can work with them quite easily under a microscope. And lo and behold, if you inject the nucleus from a somatic cell into a enucleated xenopus egg, you would end up being able to grow a fully functioning adult frog. And it would follow from this that adult somatic cells do in fact contain all of the DNA that you'd ever need. There's not, it's not getting deleted or changed as the organism matures. Now, this isn't entirely accurate. Sort of our modern understanding of genomics tells us that transposable elements and de novo mutations do technically change the sequence of somatic cell DNA. And we're going to find out about that more as we go forward. But for the most part, any cell in your body has all of the DNA it would ever need to produce a new, fresh copy or a fresh organism. A clone of you, essentially, is what it would be if you just took your somatic cells out. So DNA isn't being erased as you develop, which is, I guess, probably seeming like a pretty obvious point, but it had to be, discovered like everything else. So let's get into, and so this, I think that sort of sets the stage for the concept of epigenetics. We know that DNA is the same in every cell, yet somehow genes can be turned on and off to allow cells to perform their specific roles, not express all kinds of genes that they don't need. That's where epigenetics comes in. That is what epigenetics enables. It's a layer of information on top of the genome that regulates how the genome actually functions and what genes end up getting expressed and in how much, or in what quantities and at what times. Think about all that kind of that stuff I was talking about, the different ways that expression is regulated or the different goals in regulating gene expression. So now we're going to kind of go through and describe a variety of epigenetic marks or a variety of epigenetic mechanisms that play a role in regulating gene expression in this way. And the first one that I'm going to tell you about is the one that I'm the most familiar with because it's what I study professionally, is DNA methylation. So DNA methylation is a stable mitotically heritable epigenetic mark. What this means, and what I mean by stable, is that when DNA is methylated, when a cytosine gains a methyl group, this is effectively permanent unless it's removed on purpose. It'll stay attached to the DNA in that position, and it'll also be copied mitotically so that every daughter cell of the cell that you started with is going to carry that same exact methylation mark in the same place. This is in contrast with things that we'll hear about later, like histone modification, which the mitotic heritability of it is sort of debatable. We're not really sure if it is or it isn't, or if it is, how it would even work. And we're also much less convinced of its stability because histone modification can change rather dynamically. So DNA methylation occurs mostly at CpG dinucleotides in the mammalian genome. CpG simply refers to a cytosine followed by a guanine in the DNA sequence. The P stands for the sugar phosphate bond that connects the two. So it's not a CG base pair; it's a CG in one strand. And it's established by the de novo DNA methyltransferase enzymes DNMT3A and DNMT3B. These work, the main difference really is in what cells they tend to be expressed in, but they work to take methyl donors like acetylmethionine, SAM, and attach the methyl groups onto the 5' cytosine position. So this is so sometimes you'll call it 5-methylcytosine because it's in the 5th position on that ring. So 5-methylcytosine sometimes is also called the 5th base because you think of the 4 bases of ATCG. 5-methylcytosine is sort of an extra base on top of that because it can function differently from cytosine and it carries more information than just the regular cytosine. Methylation is copied mitotically by DNMT1. This is described as a maintenance methyltransferase. So DNMT1 can copy DNA methylation as the DNA is being replicated. It can't create new methylation, but it can copy the methylation that you already have. So these two enzymes working together kind of enable the stability of DNA methylation, because the de novo methyltransferases DNMT3A and 3B add the methylation, and then once it's there, every time a cell divides, it's copied by DNMT1, meaning that it will be stable across many, many generations of cells. Now, it's also possible for it to take place at loci that aren't CPGs. This would be called CH methylation. H is in the IUPAC nucleotide codes. It just refers to, it's sort of a wildcard code referring to either A, C, or T. And you kind of see in the literature, people have begun dropping the P. Back in the olden days, I guess, when everything had to be defined really biologically, people would call them CPGs because they were referring to a specific, sequence of DNA where C was followed by G, and not the base pairing between C and G. Now they tend to just say CH. So CH methylation is sort of the equivalent of CPG, except it happens instead of happening at a CG, it happens at a CA or a CC or a CT. So even though technically this would be called CPH methylation, that's not what it's not what people tend to call it. So CH methylation is fairly rare, but it is seen mostly in neurons and embryonic stem cells. So given that we're studying the brain, we will likely hear about CH methylation because it is happening in neurons quite uniquely. So then kind of an interesting fact, by the way, that there's a certain kind of epigenetic change or a certain type of DNA methylation that's very unique to neurons. We would believe, I think, that the brain is a fairly special organ, and things that happen that are unique to the brain must carry some information about what makes the brain so special. So this does seem to have some significance, although it's still a little bit controversial in the literature as a whole. So I want to tell you about the rarity of CPGs. Now, this, I think, could potentially come off as a fairly esoteric concept that may not be terribly relevant to the, it may seem kind of questionable relevance. But there's kind of a point I want to make here, which is that the genome is very, it's very carefully organized, and it has properties that are decidedly non-random. So if you were to just look at the human genome, say you just opened up one of those books where it's all printed out, it may look to your eyes as though the letters are all just dispersed randomly, A, T, C, G, kind of just as though you were to type those letters on the keyboard in a completely random way. But what actually happens when you analyze this more carefully is you'll find that there are distinct patterns that appear that are, like I said, decidedly non-random. So if the genome were put together randomly, there's about equal numbers of there's equal numbers of T's and A's, and there's about equal numbers of C's and G's, and the GC content of the human genome is about 42%, so there's more A's and T's than there are G's and C's. But you would basically expect there to be about equal numbers of every possible dinucleotide pairing. So there would be about equal numbers of ATs, APTs, A followed by T. There'd be about equal numbers of T's followed by A's, equal numbers of A's followed by A's, and so on and so forth down the line. There would be no reason, if the genome were completely random, for any one of those pairings to be more or less common, because you're just kind of flipping a coin every time, and it's going to give you a random distribution. So if you were to expect that, given that the GC content of the human genome is about 42%, what you would expect is that CPGs, C followed by G, would occur about 4.41% of the time. Meaning for every dinucleotide pair that you found in the genome, about 4.5% of them would be CGs. That's the expectation. But actually this is quite the opposite in real life. CPGs are very, very rare in reality. They're much more rare than you would think. So here's a figure where they're giving the frequency of all the dinucleotide combinations, and the hashed bars are the genome as a whole. The y-axis is the frequency in percent, and the x-axis shows the different dinucleotide pairings. And then the bars are colored by whether they're looking either in promoter regions of genes, so regions that are flanking the transcription start site, or whether they're looking at the genome as a whole. And what you will see is that Some dinucleotide pairings are a little bit more common than others, but what really stands out is CPG, which is all the way over here on the far right-hand side, is extremely uncommon. So across the entire genome, they're only present at about 1% frequency. And this is in contrast to the expectation that they'd be about 4.5%. So they're depleted, you might say, by about fourfold. In promoter regions, the black bars, they tend to have, they tend to be a bit more toward where you'd expect for the distribution. That's important because promoter regions tend to have a lot of CGs in them. But anyway, so they're massively depleted in the genome as a whole. And the only place where you do tend to see a lot of them, is in places called CPG islands. So these are small areas of the genome, about 300 to 3,000 base pairs long, that have many more CPGs than you would expect based on the genome-wide frequency. So CPGs are fairly rare throughout the genome, but in specific places called CPG islands, they are very, very common. And CPG islands are located throughout the genome, but they're especially concentrated in the promoter regions of genes, regions near the transcription start site. That, by the way, is why the black bar on the CPG category here is fairly high, because CPGs are rare genome-wide, but they're enriched around promoters. And the other unique property about CPG islands is that while CPGs in the genome are typically methylated, they usually have methylation there, CPG islands, usually their CPGs are fully unmethylated. So Alexis asks, why would the mammalian genome have fewer CPGs than the genome in other species? That's a good question. Yeah, so we're going to get into that a little bit in the coming slides. There is the reason essentially is that CPGs are especially easy to mutate, and so you see less of them because they mutate more frequently. I know you said that in last class you were saying that you can tell the habitat of some species based on their GC content. So thermophiles have more GCs in their DNA. So humans don't need high. That's right. So the GC content, which just refers to kind of the bulk number of Gs and Cs in the genome as a whole, does have something to do with the habitat. So animals or creatures that live in very hot, punishing environments need a lot of Gs and Cs, so their DNA doesn't melt apart. Human beings and terrestrial mammals, they don't need any more than average. So they have less than half. That's just the reason. I think there's probably a bit more to it than that. But bear in mind that GC content is different from CPG content. GC content refers to just the raw numbers of Gs and Cs in any order. So for example, here is anything that's GPT, TPC, GPA, anything with either an A or a G in it contributes to the GC content, whereas CPGs refer very specifically to the dinucleotide pairing of a cytosine followed by a guanine. So Natalie asks, could you go over the difference between CG, CPG, and GC? Losing track. Yes, of course. So sorry, this is a little bit confusing because there's just a lot of letters getting thrown around here. The basic idea is, so CPGs, that refers to in the sequence of the DNA when you have a C followed by a G. That's all that means. And anytime you see like APT, it means a dinucleotide pairing where an A is followed by a T. Dinucleotide pairings, by the way, are just one way you can divide up the genome. If you recall from your molecular biology labs or classes, you would hear about codons, right? Codons would be trinucleotide groups, the 3 nucleotides in a row that would code for a specific amino acid. So you can kind of divide the genome up into pairs of nucleotides or triplets of nucleotides or quadruplets or whatever length you want, and you would find that particular motif would appear at a certain frequency throughout the genome. So in this case, the dinucleotide pairing, there's 16 possible pairings. When you look at trinucleotide pairings, there's 64 possible trinucleotides, and the number of possibilities gets bigger and bigger the more pairs or the more you add on. So that's what a CPG is, and GC content, like I said, just refers to the kind of bulk number of guanines and cytosines in the genome as a whole. It doesn't refer to what order they are in the genome. If you have a G that's base paired with like a C, or sorry, I mean, if you have a G that's followed by a T, that counts toward the GC content, just as if you had a C followed by an A. So the CPG is a very specific situation where the C is followed by a G in the code. So here's a view of the genome browser showing the gene FOS. This is a immediate early gene that's activated in neural activity. And the green bar in the genome browser is a CPG island, and it's located right across the promoter transcription start site flanking region of the FOS gene. And this is fairly typical. When you do your genome browser assignment, I do ask you to go look for CPG islands. And once you've done what you're supposed to do for the assignment, you can go in and look kind of on your own time and see that like these do tend to localize around gene transcription start sites. That's just sort of where they tend to be located. So why are they rare? Well, as it turns out, CPGs mutate at a much higher rate than other dinucleotide pairings. So this is kind of one of these funny aspects of just looking at the genome and looking at the data. If something is absent, or if something's missing, or something you're seeing way less of it than you would expect, one of the expectations would be that something is actively removing it. If you go to the store and you're looking for a certain kind of cereal and one box is completely missing from the shelf, the assumption would be that that box is especially popular and a lot of people are buying it, or something is making that disappear off the shelves. And similarly, when you're looking through the genome and you're looking for CGs and you see way fewer of them than you're expecting, you might guess that there's some kind of process that is actually removing these from the genome, especially when you can look at other organisms that are, not mammals, for example, and see that they have a lot more CGs than we do. Where do those all go? The answer is that these CPGs mutate at a much higher rate than other dinucleotide pairs. And this is because DNA methylation happens mostly at CPGs, and methylated cytosines can spontaneously deaminate to thymine. So methylated cytosines are very easy to mutate. And when that happens, because they deaminate to thymine, the DNA repair enzymes can't find where it's happening. They just say, okay, well, this is now a thymine, okay, fine. So they don't repair it. Now, unmethylated cytosines, they deaminate to uracil, and that is something that the DNA repair enzymes can pick up because uracil doesn't belong in DNA. So that the enzymes would come along and find a uracil and just pull it out and put a cytosine back in. This is literally how DNA repair enzymes work. Is they kind of just scan along the genome for things that don't make sense, places that the base pairs don't match up, or where there's a base that's wrong or something like that. They'll come in and take out the broken piece and put a fresh one in. So unmethylated cytosine doesn't really mutate, but methylated cytosines do. So places where there's not a lot of methylation at the cytosines tend not to mutate. And as I said, CPG islands don't have a lot of methylation. They tend to be very low levels of methylation. But all of the other cytosines tend to have fairly high levels of methylation. So what you see basically is that methylated DNA over evolutionary timescales tends to be mutated where the cytosine is converted to a T, and then of course the guanine is converted to an A just to kind of complete the base pairing. So these mutations are overrepresented in human monogenic diseases, which we'll get into a little bit. But basically, the SNPs that contribute to diseases have more of these mutations at CG sites than you would expect. Does that make sense? So the idea would be that, theoretically, mutations could happen anywhere in the genome. There would be no reason why one particular position would be mutating more often than another, you would expect. But in actual fact, certain places are more vulnerable to mutations than others. That being the case, mutations that cause diseases tend to occur in certain positions more often than others. And when you ask what positions those are, they tend to be CPGs. So CPGs are more frequently represented in disease-causing mutations than all of the other kinds of mutations. Of course, TG mutations can happen too. All kinds of other mutations can happen. It's just that relative to what you'd expect, the CPG mutations are a lot more common. So there is kind of an architecture, I guess, to the genetics of disease. Throughout the genome, there are rates of mutation and frequencies of certain patterns and motifs that you would expect, and then there's what you actually observe. And this difference between what you expect and what you observe tells you a lot about what's happening underneath at the biochemical level. So when we talk about DNA methylation in the genome, we typically describe it as percent methylation. The way we calculate this is beginning with a process called bisulfite conversion. This is when you take DNA that you've extracted and expose it to sodium bisulfite. This is a chemical that is quite harsh, it's quite damaging to the DNA, and what it basically does is it converts unmethylated cytosines to uracil, which then through one cycle of PCR converts the uracil to thymine. So it essentially, this gives you something you can sequence, and you can compare the sequence you obtain to the reference DNA, and wherever you see a thymine where there should be a cytosine, you know that was an unmethylated DNA. So I don't want to get you too bogged down in the technical aspects of this. I think bisulfite conversion is not something I'm going to be asking you to explain on an exam. But I think if you can extract one principle from this, the principle that I would like you to focus on is the fact that by exposing the DNA to this chemical modification, you convert epigenetic changes, the DNA methylation, you convert it to a difference in sequence. You basically, you convert unmethylated cytosines to thymines, and that creates a sequence difference that separates methylated and unmethylated DNA. Because you can map these sequences onto the reference genome, you can tell how many of the DNA strands you had in your input were methylated and how many were unmethylated just by counting up the number of thymines you see relative to the number of cytosines you see. And then from that, you can compute the percent methylation. So here is a what we would call a lollipop or a tongue hulu plot. This is sort of how DNA methylation is represented in the genome, or in like kind of figures of the genome. So each of these lines represents a strand of DNA coming from a particular sequencing read or a particular cell. Every circle represents a CpG site where methylation could happen. If the circle is filled, it means that CpG, the cytosine there, is methylated, and if the circle is empty, it means the cytosine is unmethylated. So you can take all of your data and say, okay, in this particular position, we've got 3 methylated cytosines and 1 unmethylated cytosine. That means that position is 75% methylated. The next position down, we've got 2 that are methylated and 2 that are unmethylated, and that would mean that the position is 50% methylated. And you can kind of go on down the line and make your calculations that way. So if I'm talking about an area that's fairly high methylation, what it would mean is that about 75% or more of the cytosines in that position are methylated, whereas low methylation could mean that none of them are methylated, or maybe 1 or 2% of them are methylated. Does that make sense? So that's the main type of data that we're working with when we're talking about DNA methylation as an epigenetic change that we can study. So as I said, CpG methylation is mitotically heritable. This means that it can be copied faithfully between cell divisions. And this is really kind of a big deal, because this is exactly what enables epigenetic mechanisms like DNA methylation to carry information between generations. As we'll see when we learn about developmental programming, people are very curious about whether a trauma or something inflicted on one generation can be passed on epigenetically to the next generation. And now, of course, there's cultural mechanisms for passing on information. There's a lot of ways that this could happen, but epigenetics is sort of thought of optimistically as a way to bring this down to the level of biochemistry. If you can inherit epigenetic changes from your parents, for example, then you might inherit characteristics that they have that go beyond simple genetics. This is a way that the environment can be heritable in addition, so it's a way that nurture, I suppose, could be heritable as well as nature. So it kind of, a little bit blurs the lines between this kind of traditional dichotomy of, genetic changes that are heritable and environmental changes that are not heritable. And it says maybe some environmental changes actually are heritable. So that's what we'd like to learn about. But in order to kind of address that in a serious way, we have to learn about how is it that this even works? How is DNA methylation mitotically heritable? The way it works is basically like this. Because methylation occurs mostly at CpGs, that is a C followed by a G, the reverse or the complementary strand to that is always going to be a G and a C, because, of course, C base pairs with G and G base pairs with C. So wherever there's a CG, you're guaranteed to have a CG as the reverse complement strand as well. This is because, or the way you would describe this is you would say that CPGs are palindromic. A palindrome is like a sentence or a word you can read that says the same thing whether you read it frontwards and to back or back to front. Like, can anyone think of a palindrome? I'm a little stuck, but anyway, there's sentences out there that are palindromes that say the same thing forwards and backwards, and CPGs are like that. Not everything is like that, but CPGs are. What this means is that when the DNA is being copied and you take the two strands apart, each strand has a methylation on it. The strand, the forward strand has a methylation, and the reverse strand also has a methylation. When the DNA polymerase comes along and fills in the replica strand, Anna, yes, exactly. The name Anna is a palindrome because if you say it forwards, it's Anna, and if you say it backwards, it's also Anna. I think there's longer, fancier ones too. But anyway, that's the right idea. Dad, exactly. Yeah, that kind of thing. So CPGs are kind of like the name Dad or Anna. They read the same forwards and backwards. So what this means then is that when you copy the DNA, you have the parent strand, which is methylated, and then you have a replica strand that's added on, and then you've got a CPG where you've got a CPG on both sides. The DNMT1 enzyme, this is the maintenance methyltransferase, then comes along and copies the methylation. Taco Cat, there you go. Yeah, so this is we're seeing a lot of good names or a lot of good palindromes coming out now. This is the idea. So CPGs are like Taco Cats, or race car. Yeah. So anyway, gotta stop reading those; they're getting distracting. So. Anyway, you've got a new CPG that you've added to the other side, and the DNMT1 enzyme is designed to come along and look for places where the DNA is hemimethylated, where there's methylation on one strand but not the other, and all it will do is simply copy the methylation from one CPG on the one side to the CPG on the opposite side, and thus ensure that the DNA is methylated on both strands. This is the magic of CPGs. Because they're palindromic, you're always guaranteed to have one on one strand and then another one on the opposite strand. And all that has to happen is that information is copied over by DNMT1. This enables information to be transferred to daughter cells, and it enables the mitotic heritability of DNA methylation. Now, those are some basic facts about DNA methylation as an epigenetic change. Now, what is the purpose of all of this? It's, the way it works is somewhat complicated, and it will certainly take some time to kind of get this, I think, to kind of work through all the implications of this. But maybe it'll help you to understand what it's for while you're trying to learn about how maintenance methyltransferases work and so forth. So DNA methylation plays a role in regulating gene expression. The methylation level in the promoter regions of genes tend to be negatively correlated with gene expression. What this means is that typically, the more methylation you have at a promoter, the less that gene is expressed. This works in part because methylated DNA can alter the binding of transcription factors as well as the enzymes that modify histones. So think about all the stuff I was telling you about transcription factors and how important those binding sites are. Well, some of those binding sites have CGs in them, or CPGs, and thus the methylation level of that binding site can affect whether or not the transcription factor binds. If you affect whether or not the transcription factor binds, you affect whether it's able to cause a change in gene expression. So by methylating the DNA, you can affect the way that the whole transcriptional machinery is able to bind and whether or not it's able to express the gene. And like I said, typically the more methylation you have, the less that gene tends to be expressed. So Natalie asks, so the DNMT1 ensures equal methylation on both strands when there is heavy CPG methylation on another strand. That's right, yes. So the DNMT1 makes sure that when you copy the DNA, to put it in a nutshell, what DNMT1 does is when you copy the DNA, it copies the methylation as well. That's kind of what it does in a nutshell. DNA polymerase is what copies DNA. DNMT1 comes in afterwards and copies over the methylation as well, so that the methylation is preserved across cell divisions. So DNA methylation is, like any epigenetic mark, cell type specific. So this I want to kind of bring back into context with what I was telling you earlier about, I guess, what was it, the mutation and polymorphism lecture or something like that. Do you remember how I was telling you about MQTLs and EQTLs and how important it was to look in the correct cell and the correct tissue in order to understand what's happening? Every cell expresses different genes. Therefore, every cell expresses or has different, epigenetics. The DNA methylation is going to be different between cells because DNA methylation is there to regulate gene expression, and different cells have different genes that they express. It also, in addition to this, regulates imprinting. We'll hear about that, I guess, in the next lecture, as well as X inactivation, as well as silencing of retrotransposons. So all three of these things we will hear about in due time. I think X inactivation we'll talk about today, but the other two we'll save those for later. So it's involved not just in expression, but it's also involved in other sorts of important aspects of regulating the genome. And DNA methylation is established early in life, and it tends to increase as cells become more and more mature. So when you first start out, the methylation level in your cells is very, very low, and as they differentiate and as they become more mature, the methylation becomes higher and higher. So this is sort of a diagram of how that looks in practice. Beginning with your life, you're immediately following conception, at least, you're a zygote. And by the time you reach the blastocyst stage, your somatic cells have very low levels of methylation, less than 25% on average. However, as you go through embryonic life and begin to differentiate, the methylation level gets higher and higher until it reaches, by the time you're about 8.5 days old, it reaches a state that's fairly highly methylated and roughly equivalent to what it will be in your somatic tissues for your entire life. It kind of between 75 and 80% in most cells and in most places. The germline, on the other hand, has its epigenetic state erased. So the germline, of course, comes from your own tissue, and because it's sort of a multipotent cell type that can be converted into anything, the epigenetic state there has to be erased to make them kind of a blank slate. So the eggs end up having very low level of methylation for females who carry eggs, and the sperm ends up having fairly higher, relatively higher methylation for whatever reason. And when they're fertilized and you form a zygote, the methylation is erased from both the DNA from mom and from dad, and kind of it's almost like reformatting the hard drive, right, and erasing it back down to zero. So there are these kind of cycles of methylation and demethylation that occur over the lifespan. The only exception, by the way, to this erasing process is at imprinted loci, but we'll hear about that later. So not a lot to really worry about here. Just to be clear, part of development involves, wiping the slate clean for your epigenetics and rebuilding your epigenetic state from the ground up. This is part of what goes on during development because, for the most part, you don't want to have a bunch of extra information coming through your epigenetics that's not relevant. So let's learn about X inactivation. This is relevant to something I mentioned when it came to copy number variation, somewhat relevant. Ordinarily, all of your autosomes come in two copies. You have two chromosome 1s, you have two chromosomes 2, et cetera, et cetera, et cetera. The exception would be, of course, trisomies. For Down syndrome, for example, is trisomy 21. That means you have an extra, a third copy of the 21st chromosome. That tends to be rather pathological. That doesn't, lead to the best kind of outcomes, at least biochemically speaking. So the number of chromosomes you have is fairly tightly controlled. You don't want too many of them. And so normally you would only have 2 of each. However, the X chromosome is an exception because depending on your sex, you would either have 1 or 2. Females, or individuals of the female sex, have 2 X chromosomes, whereas males only have 1. This is, roughly speaking, there are variations in how the sex chromosomes play out, and of course sex and gender are 2 separate things. But speaking from a strictly biological standpoint, if you have 2 X chromosomes, that would tend to point you in the female direction, whereas if you have 1 and then also a Y chromosome, you would tend to be kind of directed in the male sex direction. So this creates an issue for gene dosage because, as I said, the number of copies of every gene you have, whether it's because of the number of chromosomes you have or because of the number of copies of the gene you have on each chromosome, affects the level of expression. If you have more copies of a gene, everything else being equal, you'll express that gene at a higher rate, which would mean that whatever that gene does, whatever that protein does, is going to happen at a higher rate. This This is kind of not ideal because your cell's biochemistry is supposed to be kept under fairly tight control, and having a whole bunch of extra copies of an enzyme, for example, is going to throw all of that out of whack. So the question is of gene dosage. Without some kind of adjustment, females would express any gene on the X chromosome at about twice the rate of males because they have twice as many copies of that gene. They have 2 X chromosomes. Now, the Y chromosome, you could also kind of argue something similar happens there, but the Y chromosome doesn't have very many genes at all. The X chromosome is very, very big and has tons of genes on it. So this would be quite a bad situation if all those genes were allowed to just be expressed willy-nilly. So it turns out that this process of inactivating one of the 2 X chromosomes was one of the earliest models for DNA methylation. And it was discovered by Mary Lyon back in 1961, and she was looking at it in the context of mouse coat colors. It turns out that coat color is a very good way to look at a very good way to look at X inactivation. So here's an example of it that's maybe a little bit more down to earth. We don't have as many mice running around as we do cats. The calico cat is kind of the classic example of DNA methylation and X inactivation in action. So the X chromosome has a gene that determines the color of the coat, and there's 2 versions of it. There's a black allele and there's an orange allele. And a calico cat is a female cat with who is heterozygous for the orange and the black allele. One X chromosome that she has the orange one, and the other X chromosome she has the black one. Now, you might expect, if both X chromosomes were being expressed equally, that the cat would just be a kind of uniform mixture of orange and black, kind of I don't even know what that would look like, kind of like a tarantula color, sort of like an orange and black kind of color. But of course, that's not how they look. What actually happens is that they look rather splotchy. And the reason for this is because X chromosomes are not expressed at equal levels. What actually happens is that in the embryo, or in the early embryo, every progenitor cell essentially flips a coin and determines one of the 2 X chromosomes that's going to be inactivated. That chromosome is randomly chosen, and then by way of DNA methylation, that X chromosome is covered in DNA methylation and made to be non-functioning. So the reason you see these patches is because any patch that's black would have been cells that descended from a progenitor cell that chose the black allele and silenced the orange X chromosome. And any splotches on the cat that are orange would be because the cat silenced the black X chromosome. And of course, they only express one of the 2. This is a type of mosaicism. It's a type of epigenetic mosaicism because the cells of the cat express one of the 2 different colors in a way that's randomly chosen. And because this is chosen fairly early on, it's a fairly small number of progenitors, those progenitors go on and pass that epigenetic information on to a large number of daughter cells. So all of this black splotch on the cat's back may have come from just a small handful of progenitor cells that simply chose the black allele. And by dividing endlessly and passing their epigenetic information along, that choice is preserved, and those cells kind of expand and form a big patch on the cat. So this is how that works. I think so. I read on, I don't know if this is actually true, but I read on Wikipedia that calico cats are sometimes called chat d'Espagne in Quebec, so the Spanish cat. And I think that's kind of an interesting fact because, of course, Romance languages like French indicate the gender of whatever they're talking about. So you have chat is spelled in the feminine way, and that tells you that these cats are always female. So it's one of those examples of like where a language that's not English carries more information in the same number of words than English does. So you always have to wonder, like, how does this affect how people think? There's a lot of examples of that. It's like in French, you could say like, well, if you say I spent last night with my neighbor, in English you wouldn't have any clue what the neighbor was, but in French you would know whether the neighbor was the opposite sex or the same sex, and that could have very different implications if you say you spent the night with them. So interesting fact about some of the Romance languages. So here's how X inactivation works. It's X inactivation is mediated by DNA methylation. And there is a gene on the X chromosome called XIST. It's a long non-coding RNA. Don't worry too much about what that is, but it's a long RNA that doesn't code for a protein. but it's produced by one of the two X chromosomes in a way that's essentially randomly determined. One of the two X chromosomes begins expressing X-ist, and that RNA will spread out and coat the X chromosome that it's being expressed by. So the X chromosome that's expressing X-ist, which is randomly determined, is covered in this covered in this long non-coding RNA, and then the presence of that RNA essentially attracts methyltransferase enzymes to coat all of the CpG sites with lots of methyl groups and fully methylate the entire chromosome. It's kind of an interesting mechanism because the chromosome itself basically determines whether it's silenced. If you're an X chromosome and you express X-ist, then you will silence yourself, and that's the last decision you'll ever make, is to silence yourself. It's not determined by. It's not like it's a competition between the two X chromosomes about who gets to win. One X chromosome, basically on the basis of a coin flip, decides to silence itself, and it does so using this long non-coding RNA. The result of all of this is what's called a Barr body. This is something that somebody named Barr discovered. It's a shriveled, inactivated X chromosome that's just hanging out in the nucleus next to the other chromosomes, but looks very sad and pathetic. And this Barr, whoever Barr is, I don't know who they were, but they must have noticed that female cells had these things in the nuclei, but male cells didn't. And what they are just inactivated X chromosomes. So Kiana asks, how does Turner syndrome happen? Why would these individuals have varying phenotypes given X inactivation? Right, so I think X inactivation is one of the things that makes Turner syndrome not as bad as it could be, because you do end up with a lot of X inactivation kind of protecting the individual from the really bad effects that you would have from that. But yeah, that's part of it. I mean, some parts of the chromosome, they're called they escape X inactivation, so that they manage to express their genes anyway. Plus, also just the physical presence of an unusual number of X chromosomes can be quite damaging. But really, the fact that aneuploidies of the chromosomes, or the sex chromosomes, don't tend to be quite as damaging as aneuploidies of other chromosomes is kind of really part of the whole X inactivation story. If you had, for example, autosome inactivation, and the cells were clever enough to inactivate any extra copies of the autosomes, then Down syndrome probably wouldn't even exist. You would just have cells identify, oh, there's an extra 21st chromosome here. Let's inactivate one of these, and then you only have two. That would be a very, if cells could do that, then there wouldn't be any worries about chromosomal abnormalities. Okay, so let's just take a little bit of a breather and give you some basic facts about DNA methylation in the brain. So I'm going to speak about this in rather general terms because I think you've heard enough about the really technical side of DNA methylation, at least for now. We're only halfway through the lecture, but we'll see. So here's a basic fact about DNA methylation. It increases in the mouse and human cortex with age, and it increases in the brain as a whole. So in this figure, which comes from Lister and company, you have the y-axis represents methylation. On one side, we have CH methylation. I remember I told you that only happens in neurons. And then on the right-hand y-axis, you have CG methylation. So it goes from 0 to about 80%. Then on the x-axis of this plot, you have age in humans, birth, 0 days, all the way up to 20,000 days. I don't know how many years that is. I guess it's like 55 years old or so. So you have age on the x-axis and methylation percent on the y-axis. And what you will see is that methylation levels go from basically are fairly high at birth and just get a little higher. Like, this is like a bad scale, but like you can see the methylation levels in the human brain go up right around childhood and birth, but not by a whole lot because they're already fairly high. But they do go up significantly and then remain high through most of your adult life and begin to decline with age as your neurons begin to break down. Around about the same time you see this gain in methylation, you also have a gain in the density of synapses. So this orange line represents the number of synapses, and at the same time as methylation is going up in both the CpG and the CH positions, there's also an increasing number of synapses at the same time. So they would argue that this is not a coincidence. DNA methylation regulates neural maturation, and as the methylation goes up, the number of synapses also goes up, and this plays a role in just sort of synaptogenesis and the ongoing process of maturation. Also, potentially why the brain is so vulnerable at this time is because its epigenetic development is happening at the same time as synaptic development. The levels of the de novo DNA methyltransferase DNMT3A peak sort of midway through the first part of the postnatal period in the mouse brain and decline by adulthood. So most of the de novo methylation in the brain is happening prenatally and in the early postnatal period. This is when the cells are kind of setting things up. It's like, you know, when you're born, your cells are there, but they're not fully matured yet and they're not fully differentiated. So it's a bit like, you know, if you're building a house, the last thing you do is you come in and you put in the baseboards and you paint the walls and you put in the light fixtures and these kind of finishing touches. This is what's happening during the early postnatal period, is your neurons are kind of going through the epigenome, adding in methylation wherever they need it, and putting the finishing touches on the epigenetic state that gives them the mature and stable levels of gene expression that we would need to be functioning neurons. So this is just a Western blot showing the levels of DNMT3A relative to the control gene GAPDH in a variety of organs. And when you're looking at a Western blot, basically the darker the splotch, the more of that protein is being expressed. Westerns measure protein levels, right? So here in the cortex, for example, you see a really dark splotch for 3A at postnatal day 2, P2. And then you see a somewhat lighter splotch at P14, postnatal day 14, and then you barely see anything in the adult. What this means is that the protein is expressed at very low levels in the adult mouse brain. Because at that point, all the methylation has really been established and it needs to do, you know, there's comparatively little work that needs to be done to set that all up and to add any more methylation. And then the expression of DNMT3A expressed in the mouse is peaking postnatally for mRNA, but the DNMT1 enzyme declines from the moment of birth. So this kind of makes sense as well, because DNMT1 is involved in copying methylation across cell divisions, but in the brain, cells more or less don't divide after birth. You kind of are born with a certain number of neurons, and with the exception of adult ongoing neurogenesis, which is a pretty small percentage of your neurons, most of your neurons are done. They don't need to divide anymore, and so they don't need any more DNMT1. DNMT1 is only there to carry methylation across cell divisions. So neurons have a lot of DNMT3A in the early postnatal period, but not very much DNMT1. Now, here is a fun picture. So this is kind of what it looks like in practice, what DNA methylation looks like in practice. So what we have here is kind of a genome browser view. You would be familiar with it from the UCSC genome browser assignment. What we have are the gene MEF2C. And so you can see the gene is coded on the negative strand. It's pointed this way. It's pointed from right to left, and then it's located on chromosome 5 between these two coordinates. Now, in the genome browser, you can add tracks that carry other types of information on top of just the genetics. In this case, we have a track carrying information about DNA methylation. And where you see a lot of green, that means that there's a lot of DNA methylation. The methylation is at a very high percent. And where you see not very much green, what it means is that the methylation is at a lower percent. Now, remember that I told you that methylation is negatively correlated with expression. So the more methylation you have in a gene, the less that gene tends to be expressed. And the less methylation you have, the more that gene tends to be expressed. MEF2C is a transcription factor that's expressed in neurons. So what do you see when you look at this? Well, in the NEUN positive tracks, NEUN positive being a neurons, NEUN is a marker of neurons, you'll see that this gene has fairly low methylation across the entire gene body. You can tell because of all this extra white space. NEUN negative cells, which are not neurons from the brain, there would be like glia or something, tend to have very high methylation across the gene body. So this is just an example of what you could see on the genome browser. If you look at levels of methylation, you can actually see how they play out across the length of genes and across other features that you might be interested in. And typically, what you see is that in genes that are highly expressed in a certain cell type, they tend to have low methylation, and you can observe it in the genome browser compared to genes to cells that don't express it. Okay, so Yuma asks, would you be able to go over again why neurons have DNMT3A again? Yes, so to put it very simply, DNMT3A is a de novo methyltransferase. It's responsible for adding DNA methylation to DNA and setting up the epigenetic state of those cells. So this happens fairly early on in life. Once the epigenetic state is established, it doesn't need to be, you don't need to keep having this enzyme there. So it's expressed early on in life while the cells are setting themselves up, while they're putting up all the DNA methylation that they need to maintain gene expression. Afterwards, the levels tend to decline. Adult brains don't tend to express very much DNMT3A. Now, what I'm trying to emphasize is that there's a contrast between DNMT3A, which is the de novo methyltransferase, meaning that it adds new methylation, with DNMT1, which is a maintenance methyltransferase. The maintenance methyltransferase is only there to copy methylation across cell divisions. And in the brain, neurons don't really tend to divide after the fetal period. So DNMT1 becomes very low. DNMT3A remains high a little bit longer, but ultimately by adulthood, if you look at this plot with age, by the time the mouse is about 60 days old, both the DNMT1 and 3A are at fairly low levels because most of the action for DNA methylation has already been taken care of, and because the DNA methylation is very stable, almost kind of permanent, once it's all taken care of and established, you don't need to go in and keep working on it. It's just there and you're all set for life. So let's go over another example of DNA methylation in action. We've given you X inactivation. This is one way that DNA methylation is involved in things. But here's another kind of fun example. This is the agouti viable yellow mouse. So this is a type of mouse that was discovered basically by accident. If you breed enough mice, you'll get all kinds of weird and wild mutations that come up, and you can look at them and see what they do. Typically the ones that affect the coat color are what you would notice. But basically this is a mouse where you can have genetically identical twins or litter mates that differ substantially in their coat color. These mice could either be yellow, and they're very, very cute little guys when they're yellow, or they could be brown. The yellow would be the agouti color, or so the yellow color is one option. They could also be brown, or they could be anywhere else in between, mottled. They kind of have patches of yellow and brown color. Now, the reason this happens is because of a mutation upstream of the agouti gene. There's a gene called agouti that basically makes hair turn yellow. And when mice have this mutation, they become all yellow. Normally, the agouti gene is only supposed to be turned on a little bit to help make the fur brown. When you combine black and yellow, sort of makes it brown. So normally the agouti gene is not supposed to be on all the time. But in the yellow AVY mice, the agouti gene is turned on all the time. But here's the beauty of it: the level of methylation at this mutation, this intracisternal A particle, just call it the IAP, this mutation, determines how much of the agouti is actually expressed. So if you have a lot of methylation at this mutation, it means that even though the mutation exists, the mouse won't be yellow; it'll actually be brown or pseudo-agouti. So the punchline here, I don't need you to worry too much about what an IAP retrotransposon is or any of that kind of stuff. The basic message is very simple. The methylation level at this particular spot in the mouse's genome can allow you to predict with almost complete certainty whether the mouse is going to be yellow or brown, or orange or brown. These are mice that are genetically identical. They're twin sisters essentially. They have no genetic difference, yet their coat color and how fat they are and how healthy they are is determined by the methylation level at this one particular gene. So this is called a metastable epiallele, and it's a favorite. It's kind of an interesting topic for a number of reasons, but people are very tempted by the fact that this methylation has such a huge effect on the mouse's phenotype, despite the mice all being genetically identical. Because you could ask, well, are there things like that in humans? What if there was a place that you could measure in the human genome, or what if there was a place where you could measure methylation in humans and tell with complete certainty whether, for example, you were going to develop cancer? Wouldn't that be a great thing to find out? So people look at these mice as a very interesting example of methylation in action, showing just how powerful differences in methylation can be. So, to put it in more concrete terms, these are the agouti sisters. On the left-hand side, you have the mouse pups, basically the day they were born. You can't tell what color they are because they don't have any fur. And then here are the mice grown up. The one on the left hand became this big round orange character, and the one on the right became kind of a normal-looking brown mouse. These two are genetically identical. The only difference between these two is the level of methylation at this agouti locus. So one little spot, if you measure the methylation there, you'll tell with complete certainty what color the mouse is going to be even before you can tell the fur. If you were to just take a drop of blood from both these mice, you would know which was going to be the yellow one and which was going to be the brown one. And like I say, the dream is always to find a version of that for other things in humans. Imagine if you could look at one place and measure the methylation there and find out whether somebody was going to be at risk for cancer or for schizophrenia or some other kind of serious disease. That would be a pretty major breakthrough. So this is an example of what DNA, that sort of the power and the potential of DNA methylation. Now, let's get on to another topic. This is about chromatin biology. So I think I referenced this a little bit in the last class. The DNA is fairly carefully organized. It's not allowed to just flop around endlessly in the nucleus. It has to be tied up. Natalie asks, just curious, it seems that the agouti mice are larger in size than the brown mice. Is there a reason for this? Yes. So I don't know if you've ever taken one of the neurobiology of energy homeostasis, one of the like alpha-abazade classes. You might have heard of AGRP, agouti-related protein. This is a gene that's expressed by your hypothalamus, and it makes you hungry, in a nutshell. So the agouti gene and genes related to it not only turn your coat yellow, but they also, in your brain, make you very, very hungry. So these yellow mice express more of this protein and thus eat more and have, end up being fatter than the regular mice. So there's a number of sort of phenotypic changes. The coat color is the most obvious, but there's also all these metabolic changes as well. That's the reason for that. So anyway, so the DNA is very carefully organized. It's and without getting too much into the technical details, it's wound around these things called nucleosomes and packed away in a very well-organized bundle inside the nucleus. And I was thinking, it's kind of analogous. I mean, this image to the right-hand side shows what people used to do when earbuds had cables. You would wind them around these little kind of mandrels, and that would keep the cable from getting all tangled up in your backpack. Now, of course, we all have, the AirPods that don't even have cables, so this is kind of redundant. But back in the day when you had cables, you had to worry about this kind of thing. So this is what's going on with the DNA. The DNA is wound around these nucleosomes that are made up of histone octamers. You have H1, H2A, H2B, H3, H4. This is one of those times when you're going to be happy that the quizzes are all open book, because you can just always look this up if I ever ask you what are the different kinds of histones. You don't have to memorize all these different letters. Although I don't think I'd ever ask you that, because it's not, for our purposes, it's not super important. Anyway, so the DNA is wound up very carefully, and this is how it's put together in the nucleus. And histone modifications are a type of epigenetic change that affects these histones and how they interact with DNA. So it's possible to take the N-terminal tails of the H3 and H4 proteins and add different kinds of things to them that change their relationship with the genome or with the DNA. You can do things like add acetylation, you can add methylation, you can add ubiquitination and all kinds of other things. There's many, many to name, and we don't want to get too bogged down in this. But what they all end up doing is modifying how the histone protein interacts with the DNA and with the local chromatin structure. So roughly speaking, if you acetylate or phosphorylate histones, that tends to give them tends to make them more negatively charged. Now, DNA itself is negatively charged, and so if you have a negatively charged histone and the DNA itself, which is negatively charged, they tend to repel each other. This basically loosens up the DNA. It's kind of like when you take your shoelaces and kind of pull them loose a little bit, all the loops get less tight. This tends to make DNA more accessible to transcription factors. If the DNA is all bundled up in a big kind of wad, it's very tough for transcription factors and RNA polymerase to get in there and begin expressing genes. So histone modifications like acetylation and phosphorylation loosen up the DNA and loosen up the chromatin, making it easier to express those genes. So when we talk about histone modifications, the way the nomenclature works is they're described in terms of what histone are we talking about, what amino acid in the histone is being modified, and then finally what the modification actually is. So this is kind of just a technical note, but there are Like, let me give you an example of how these things are talked about, and then you can come back and look at this to reference anything else that you see. So let's say we're talking about H3K4ME3. That means we're talking about histone H3. That's one of the types of histones that exists. We're looking at K4. That's the lysine. K is the symbol for lysine. So it's the fourth lysine residue in the histone tail. And then Me3 means trimethylation. It means you've added 3 methyl marks onto that. And this is just one of the many, many different kinds of modifications that exist. I'm certainly not going to ask you to memorize them all, but this is just the nomenclature of it. You should be able to look at that and think, okay, so this is an H3 lysine modification kind of thing. And but incidentally, this is something that's involved with promoters of actively transcribed genes. I'll tell you a little bit about that in the next couple of slides. So histone acetylation, as I was saying, tends to relax or loosen up the chromatin, leading to increased gene expression. So chromatin can kind of be in different states of relaxation. You can have it very packed together, and the genes that are located in packed together regions of the DNA or of the chromatin don't tend to be expressed very highly. So that's a picture of this here. All the chromatin, the DNA is wrapped around these nucleosomes, and it's all kind of packed together very tightly. But if you add acetyl groups to some of these histone tails, it tends to spread out the DNA and create these open regions between the nucleosomes that make it easier for the enzymes and whatnot to get inside and bind to the DNA. So what you're ultimately doing with these modifications is changing the three-dimensional kind of arrangement of the DNA or of the chromatin such that it makes certain regions and certain genes more or less accessible to transcription factors and enzymes. So getting back to H3K4ME3, this is, again, just purely an example of what these types of things look like. It's not something that you need to remember as a fact on its own. But as I said, this is a type of histone mark that that demarcates actively transcribed genes. So the more H3K4ME3 you have, the more actively those genes are expressed in that cell type. So this is ChIP-seq data. We'll learn about that later, but it's a genome browser image as well. So what we have are neurons and non-neurons. They've used flow cytometry to separate neurons and non-neurons from some piece of the brain, and you're basically comparing neurons to glia, and you're looking at the level of H3K4ME3. So add a gene like GAPDH, which is a sort of what we would call a housekeeping gene that's expressed by all cells. Whether you're a neuron or a glia, you need GAPDH because it's part of the Krebs cycle, so you need it to live. You'll see equal levels of this mark in GAPDH in both neurons and non-neurons. This track just shows you, across the length of this gene, it shows you the level of, I wish my pen was working, I could show this a lot better, but it shows you the level of H3K4ME3 along the length of this gene. And you can see that it's the same in both neurons and non-neurons. Don't worry about this bottom one. We'll leave that for now. But so just on the top 2 rows, you see that there's about equal levels of this across the GAPDH gene in both neurons and non-neurons. Now, compare and contrast that to the gene BDNF. BDNF is brain-derived neurotrophic factor. That's only made by neurons. So this is a gene that is not expressed by glia. And what do you see here? Well, you see a lot of this mark in the gene on neurons, but you see almost none of it on non-neurons or glia. So what this means is that this is an epigenetic mark, H3K4ME3, that can tell apart brain cell types. If I were to just give you 2 sets of cells and say measure the H3K4ME3 in these 2 sets of cells, you could tell just by looking at where there's a lot of this mark what the cell was. Genes that are highly expressed have a lot of this mark, and genes that are not highly expressed don't have very much of it. So I've given you here a table that shows all the different modifications, and I assure you I don't want you to memorize this. I just want to give it to you as a sort of a symbol, or to give you a sense of how complicated this all is and how many factors are involved here. There's all kinds of different modifications, and they all tend to have different effects. So don't worry about it too much. But someday you might hear about H3K122 acetylation, and if you were to look at this table, it would tell you, okay, genes that have a lot of H3K122 acetylation tend to be activated. On the other hand, genes that tend to have a lot of H3K9 dimethylation, those tend to be repressed. and so on and so forth. So you could kind of go through and look at all this if you wanted, but that's not really my interest. You can, keep this as a reference for yourself later. So as I was alluding, chromatin has a three-dimensional structure. The way that the chromatin is set up is not random. All of your DNA is not just kind of crumpled up and stuffed into your nucleus like it's like you're packing for school and your bus is, you're 5 minutes late for your bus. It's not like that. It's all very carefully organized. It's like one of those gamer PCs where all the cable management is really, really nice and tidy. That's kind of like what your DNA is like. It's organized into a three-dimensional structure, which not only allows it to fit inside the nucleus, but as we're discovering now, also plays a fairly big role in regulating gene expression. Neurons, being that they don't divide, spend almost all of their time in interphase, and in this part of the cell cycle, chromatin is divided into open and closed segments. The closed parts are called heterochromatin, and they're very condensed, and it's very difficult, as I've been trying to emphasize, when the chromatin is condensed and stuck together, it's very difficult for transcription factors and enzymes to access. In contrast, open chromatin, or as it's called, euchromatin, is looser, and so genes that are located in euchromatin are easier to transcribe. And these aspects of the three-dimensional structure of chromatin are mediated by DNA methylation and histone modification. So regions that have a lot of methylation and repressive histone marks tend to be heterochromatin. If you were to look at the methylation levels across the genome, you'd see that the methylation was very high in heterochromatin and much lower in euchromatin. And you can actually see this in situ. There's an example I've given you here of a cell, and when you're looking at the nucleus that's been stained with like DAPI, which is a fluorescent dye that binds to DNA, you'll see that there's very bright spots in the nucleus where there's a ton of DNA all condensed, like all sort of stuck, crammed together. Those are areas of heterochromatin, and the lighter colored areas are areas of euchromatin. And like I said, heterochromatin is where the genes you don't want to be expressed, and euchromatin is where the genes you do want to be expressed are located. Okay, so I think I'm going to skip this slide for now. This is about another type of sequencing that tells you where there are regions of accessible DNA, but I don't think we really need to hear about this right now. We'll save this for later when we actually talk about it in the context of some research. But I do want to tell you, with the time we have left, as much as I can about the way three-dimensional correlations in the genome matter. So if you recall from our last lecture, there are things called enhancers that can regulate gene expression by, even from a very long distance away from their target gene. And the way they're able to do this is a transcription factor can bind to an enhancer, and then the DNA where it's bound can fold over and loop back to the transcription start site of the target gene. And even though like in the linear view of the genome, they're a long ways away. If you were to look at them on the genome browser, for example, you'd see they were very far apart. But in the real cell, they're three-dimensionally very close to each other. So this kind of three-dimensional structure, the folding and looping of the genome, plays a big role in regulating gene expression due to these enhancer gene relationships. And one of the ways you can tell this is with a type of in situ hybridization. And basically the idea is you can use fluorescently labeled probes. I've talked about fish before, but you can use fluorescently labeled probes to mark different places on the genome. And then you'll see, when you look at those cells under a microscope, that those places glow in the dark. And so if you mark one place with a red color and one place with a green color, and you look under the microscope, you can tell how close they physically are in real life. Now, if you were labeling places that were very far away in the linear, in the linear view of the genome, but then you looked in the cells and you found that they were always very close together. I've given an example here. You can see that these red and green points are very close together. You would know that the way the chromatin is actually configured in 3 dimensions has these areas very, very close to each other. So that's just kind of to make a point that when you look at the genome under the genome browser, it looks like a big long line, and things that are very far apart, you would assume don't influence each other. Just like this, what's going on in BC doesn't really affect us here in Ontario, or in Nova Scotia, much less. But that's not the way the genome works. The genome has a linear structure, but it also has a 3D structure that depends on how it's all folded together. It's kind of like if you were like a, well, you always hear about faster than light travel working by folding space-time. Well, the genome's already figured this out. It can already achieve long distance travel just by folding itself up and bringing areas that are far apart in contact with one another. So we'll learn about the practical implications of this later. But right now, I just want you to kind of understand and conceptualize that there is a three-dimensional structure to the genome that really matters for how things work. Okay, so the last thing I wanted to show you was, again, I'm trying to kind of maybe a little bit blow your mind with this to show you how many layers of epigenetic information stack on top of the genome itself. And I'm not saying that this is something you have to learn all right now and memorize all at once, and that it's going to be like a pass-fail kind of situation. I'm really kind of just trying to expand your consciousness and give you a broader view on things. So to that end, I want to show you what a genome browser view would look like when you add all of the information we know about all on top of it, all together. So this actually came from the Roadmap Epigenomics Consortium. This was a paper done in 2015 that was basically just trying to do, it's a consortium project that was trying to catalog all of the types of epigenetic information. So here we have the very top row gives you the genes. So we're looking at genes like FAM205B, REC, SIT1, NPR2, all these kinds of genes. So we're looking at a fairly long section of the genome. And then underneath we have genome browser tracks that represent all of the different other kinds of information you can get in mouse fetal lung fibroblasts. So you've got RNA-seq that tells you how much of that gene is expressed in those cells. Then you've got all of these different ChIP-seq tracks, H3, K36, ME3. The bumps here show you how much of that is present in all of those locations. Then you've got bumps representing all of the other different kinds of histone modifications that are like histone methylation marks of H3. Then you've got DNase-seq that tells you whether the chromatin is open or closed in that place. You've got all kinds of other histone modifications. Just look down this line, like all of, I guess maybe you can't see my mouse, but anyway, like all of these tracks, or at least almost all of these tracks are just different kinds of histone modifications that are present in varying amounts in different parts of the genome. Then you've got WGBS, that's whole genome bisulfite sequencing. That's what we were mentioning earlier. That tells you how methylated the DNA is. And then you've got Hi-C that it tells you what parts of the genome are touching each other over a long distance. We'll talk about that later as well. So again, I'm just trying to kind of blow your mind and give you, to raise your consciousness about how many layers of information are layered on top of just the genes themselves. If you just look at the genes themselves, you're only seeing the very tip of the iceberg. There's so much more on top of it that controls how those genes actually function in the cells. So keep that in mind. We'll learn about select parts of this as they relate to mental illness and so forth. But this is, just as kind of an overall view of what's possible. So I'll just close by giving you a media recommendation. So the Lex Friedman podcast did a 3-part series of interviews with Manolis Kellis. They're all about over 2 hours long, but he was kind of the leader of this Roadmap Epigenetics Consortium. He's a computational biologist who studies this stuff, and he does a terrific job of explaining all of the stuff about computational biology and epigenetics and so forth. So I would highly, and they're terrific interviews. So I'd highly recommend if you have 6 or 7 hours to spare. To visit these on YouTube or in your favorite podcast app and listen to them. I think he does a terrific job of explaining it all. One of the real brain geniuses of the genomics and epigenomics world, and we're going to be hearing about his work in future lectures. I have a lot of praise for it. So, like I say, if you want to hear somebody else talk about this stuff in a more kind of lucid and slowed down way, in a more conversational kind of way, I would highly recommend these podcasts. I wouldn't recommend listening to Lex Friedman's political podcasts. I think those tend to be pretty naive, but when he talks about science or when he talks to scientists, those are quite good. So there we go, folks. I don't think we have, we're kind of running out of time when it comes to the slideshow, but if you have any questions just to kind of cap off, I'll take those now, or you can always email me if you like. All right, folks. Well, anyway, so thanks for listening. I think that was probably a fairly complicated lecture, just going through it and giving it to you. I'm like, oh boy, there's a lot. There is a lot here. So don't don't feel bad if it's coming at you pretty fast at this stage. I think what I've found from the previous times I've taught this all becomes much more real when we apply it to the actual mental health conditions. What I'm trying to give you is like an overview of the whole scope of the field. To say like, this is how big the field is. This is the whole list of all the things that can be happening inside of neurons to regulate how they function. Let's take little slices of it and look at how they influence this or that condition and illness. So we're giving you an overall scope now, and then we're going to kind of try to zoom in and focus on things in more detail as we go forward. So if that's all today, folks, thank you very much, and enjoy the rest of the class. I think I'm going to have to cancel office hours tomorrow, but I'll be available by email if you need to contact me, and I'll be back at the same time and place next week. All right, folks, we'll see you next time.